Hello, everybody, and welcome you all to the last day of this year's IABS Stats Workshop. My name is Timo Weiler, and I'm co-chairing this session together with Theo Kulis from Roche Genentech. It's great for all of us, for all of you to join us today. We have a great case study session planned for you. The theme of this session is advancing CMC statistics for a new generation of statisticians, regulatory reviewers, scientists, and modelers. I have been co-chairing this case study sessions over the last few years together with Theo. And if you have joined the IABS Stats Workshop in the last few years, then you know that I think that professional statisticians in CMC, whether statistician or data scientists, is really working as a statistical consultant. I have noticed that we have to deal with the fact that the application of CMC statistics and the respective requirements that come with it are constantly evolving over time. So it's not surprising that CMC statisticians are helping with the development of new methodologies in order for the CMC world to face these challenges and to add value in the CMC context. But with each new methodology, the CMC statistician faces a multitude of challenges. And of course, the way we learn as statistical consultants is really by sharing our solutions in the CMC world with other CMC statisticians. So this session includes case studies that showcase new applications or elucidate on the respective challenges in applying novel methodologies. We have some great speakers with us today, and I'm looking forward to hearing and learning from them as they discuss their case studies. So first of all, the session will start with the talk from Mark Gang and Gang Wang from Moderna, who will talk about continuous manufacturing and the future of mRNA production. For the second talk, Tobias Eilert from Boehringer Ingelheim will show us how Bayesian framework can be used for stability studies for life cycle products. These two talks will be followed by two parallel breakout sessions as you're used to. The topics of these breakout sessions today will be Bayesian and statistical software for non-statisticians. And the final speaker of this session is Christian Oliva-Avies from Genentech, who will talk to us about using an Arrhenius perspective for modeling stability data. The session will finish, as you're used to, with a panel discussion with all the presenters from today. So with that being said, I'm looking forward to our first presentation of the day. I think, Gang, you already turned your camera on. Thank you again. I think I will now welcome you back on stage for the presentation about continuous manufacturing. I also saw Mark in the audience. Great. Yeah, Mark is also there. You're also invited to turn off your camera. So if the two of you are presenting. So Gang is the technical lead in mechanistic modeling and machine learning at the technical development department of Moderna. And Mark has worked on COVID vaccine development, testing, manufacturing, and regulatory filing at Moderna. His responsibilities include, among others, early and late stage process development. Currently, Mark focuses on the next generation product development and digital twin applications. With that, I would like to hand over to the two of you. Let's proceed to the next presentation. If there are any more questions, we will be happy to, or you can ask them later on in the panel discussion. There will be lots of time for any open questions. With that being said, I would like now to come to the second presentation of today. Therefore, I would like to ask Tobias Eilert to join on the stage. He will talk about a modern approach to stability studies for life cycle products using a Bayesian linear mixed model framework. Tobias is a senior principal CMC statistician at Boehringer Ingelheim. He's predominantly interested in the harmonization, standardization, and modernization of statistical approaches in the CMC area. With that being said, Tobias, the virtual stage is yours.
Thank you, Timo. Then let me try to share my screen. Is it already visible?
Yes, but not in full screen. Can you go to presentation mode?
There
Great,
you go.
yeah.
Perfect. Nice. And I think also my camera is on, maybe a bit too bright, but I think you can see me. It's already getting dark here. So yes, thank you for the opportunity. Tim already introduced me. I'm happy to give a talk here and share later discussion and comments with you. Today, I will talk to you about a modern approach to stability studies for life cycle products by using a Bayesian linear mixed effect model framework. As I guess most of you know, the still global standard for stability analysis is the ICH Q1E. However, in the last years, there were many publications which criticized many aspects of this guidance. From these limitations, we developed a whole interpretation translation of this ICH Q1E guidance to a Bayesian linear effects model. With these solutions, I will only present to them in brief at the beginning. We'll pick out some for details, which we then later need for case study 1, where we show the similarity of both approaches, the novel Bayesian LMMN approach and the ICH guidance. And then in the second one, in the second case study, where we use real data, we can show them the real power of the linear mixed effect models. This then I will conclude in one slide and then open for discussion. Let me show if I can also get a nice laser pointer so everyone can see it. So as most of you know, is that the ICH Q1E suggests a linear fixed effect model for analyzing the stability data. Here you have the response for time t and batch j, and this is modeled usually with a overall intercept and an overall slope, alpha 0 and beta 0. However, in a fixed effect model, it's important to know this is not a real overall intercept. This is just one batch out of these whole batches which we simulate, and then we simulate the others or estimate the others just by deviations from them. Further, the ICH Q1E says that the residual error is normally distributed with a single standard deviation. That means they assume homoscedasticity for all times, but also all batches. From this estimation then, you get for each batch a confidence band of mean regression, as you can see here on the right. And the ICH guidance suggests that the batch shelf life of each batch from this analysis is determined by the intersection of the shortest intersection of the confidence band of the mean regression with the specification limits. Here in this formula, we kind of made it more proper, because in the ICH there is only a prosa text about that. We defined this as a supremo, here only for simplicity for a lower specification limit. Then the ICH guidance says to determine the product shelf life, so the overall shelf life, just by the minimum of all batch shelf lives. As you know, most of you know, the ICH comes further in the fixed effects approach with 3 different models: the separate intercept, separate slope, or short the cis model, the separate intercept and the common slope, the 6 model, or the most favorable one, the common intercept and common slope, the KIX model. Here you go from least favorable to most favorable, because this model usually returns the shortest shelf lives, and this returns the longest shelf lives. Here it already starts with some problems. These models, because they are based in a fixed effects approach, do not allow to include the batch-to-batch variation. And with that, we also cannot, because there's no parameter for it, we cannot estimate its uncertainty. We only have a minimum approach, as I showed one slide before. Further, the more batches you include, usually the less the shelf life gets, which of course is detrimental for practitioners because they fear, especially in a low batch regime, a low number of batches, they fear if I include more batches that my shelf life gets shorter. This is a wrong tendency. Further, we have, of course, to decide between these three models. And there the ICH Q1E suggests pullability tests based on ANCOVA F tests. Later in the examples, I will go more into detail, but here I will just give a short summary about the limitations which are given in the literature. Here, the ANCOVA F test is misused as an equivalence test with a significance level of 25% to account for a low number of batches. Further, also here, I will also get more into detail in the examples later. The more batches and measurements you include, the more it is probable to detect irrelevant differences and add up in the less favorable regimes. Further, not really detrimental limitation, but for a statistician, not so satisfying limitation, this definition or determination of the shelf life is not a real parameter, so you cannot do statistics with it. Motivated by these shortcomings, we developed a kind of a complete translation of this ICH Q1E in a Bayesian framework with a linear mixed effects model, and started off with defining customized linear mixed effects analogs. Here on the left, in analogously to the CIS model, we have the RIES model, random intercept and random slope. Then the more favorable model, the next one, the random intercept and the common slope model, the RIX model. And then at the end, the common intercept and common slope model, and the KIX model, which is exactly the same as in the fixed effect model. So we only introduced two new ones. Here, we directly addressed one problem. Here we can explicitly model the collective batch behavior. And if we have parameters about the collective batch behavior, these are parameters about the real product. So the collective batch behavior is the product behavior. With these product parameters, then we can also infer the uncertainties of these parameters, which is not possible in a fixed effect model. Further, in contrast to the ICH approach, the more batches you measure, the better gets your estimate of your population product parameters, which is kind of the direction we should go. You should motivate to get more batches. Further, the pullability tests, we switched to the Bayesian regime. Here we use the base factor for model comparison. I will also go later into details. Here, the misusage is corrected. So the direction of the model finding is corrected. And further, also here with the linear mixed effect model, it is that the more batches you measure, the more measurements you include, the more it is that you select the most fitting model. You're not scared to end up in the least favorable one. Further, also, this estimate here can be, we defined here a proper mathematical definition via the batch distribution, which I will explain now on the next slide. Here, I would like to go into detail to the two analogs, the CIX model and the RIX model, because these were chosen for the case studies. As a short reminder, the ICH has this model equation here. Just to the beginning, the beta J dropped because we have a common slope approach. However, it stays the same, that the batch behavior is just described by batch shelf life and taking the minimums of them. In the linear mixed effect model, it presents it a bit differently. We have on the first side the same equation. However, this alpha 0 here is not just one batch of all of them, but it's kind of a not existing overall intercept for the batches. So further, the alpha j here is not only the deviation from one batch to or one designated overall batch to the other, but it's really from the overall batch mean to the realizations of this distribution. And this distribution is defined by this alpha j, which is assigned to be a normal distributed random variable, where we have another parameter now, which is really important, the sigma alpha, which gives us the variability of our batch distribution. I already said it. From that, we can now define the batch distribution. This is then the random variable, which explains our random batches. And with this, we have now 3 population parameters: the overall intercept, the overall slope, and the variability, the standard deviation of the intercept variability. And with that, we can estimate and get also uncertainties of the whole batch. That means product distribution. And from this, then we can also define, similarly to the ICH, a shelf life as the intersection of the quantile of the estimated batch distribution with specification S. Here again, for simplicity, only with this lower specification limit. Because we're in a Bayesian regime, of course, this is done via the posterior predictive distribution. Okay, so after all these formulas, we will go now to the case studies, and you will see it will get really intuitive. So the first case study, the setup is that we have 6 batches. We have a lower and an upper specification limit. However, we just look at the lower one because we have a decreasing tendency. And our theoretical aim here was to extend the shelf life to 36 months. Of course, before we go into the data, into the analysis, we need to check which model we can choose. For that, in the ICH linear fixed effect model, we use ANCOVA F-tests. And for the first, the idea how it is described in the ICH is that you are in this model and you want to go to this model. However, what the hypothesis testing really does is the opposite. The null hypothesis says we have no difference between slopes, so we have a common slope. But the alternative, where we are right now, but which we kind of want to disprove, is the alternative hypothesis, which says we are in separate slopes. So kind of we're testing from here to here, but how we use it, we test from here to here. In our example, for example, here it is used, we have 36% as a p-value, so that means we have no evidence. We can go to the next step, to the next model, to the 6th model. And again, the same for the intercepts. Again, this misusage of the null, we are in the alternative hypothesis, but we want to disprove the null hypothesis. And here, as suggested by the ICH, we have now evidence, so we stop in the 6th model. In the Bayesian framework, it is a bit more intuitive. Here, as I said, we used Bayesian, the base factor. And here the base factor is used such that we are in this model and we only jump from the Rios model to the Riggs model if we have enough evidence, here given by a base factor of greater 100. So that means here in our case, we have really distinctive evidence that we should go to the Riggs model. But we do not have enough evidence to assume the Kicks model is more favorable than the Riggs model, so we stay in the Riggs model. With this, we come now to the results. Here on the left, you see the batches or the data of the batches in different colors. This the points for the data points, the solid lines for the mean linear regressions, and only for convenience, the lower 2.5% confidence limits. As you can see, we are lucky, we can make the 36-month. And for the Bayesian linear mixed effect model, we get a similar picture. We again get the data, we get the mean regression lines and the lower uncertainty bands of 2.5%. And we can also directly see they are fairly similar. However, to be honest, in the linear mixed effect model, we are really not interested in these batches here or in their estimations. You could kind of delete them and you could only look at these black curves. And these are the uncertainty bands of our collective batch distribution. That means here we have now these three parameters which I introduced before. We can with them now determine a upper and lower uncertainty bands where we think with our estimations all of the batches will lie, given the data, of course. Further, the Bayesian approach comes here with the candy. We can directly get the OOS, the out of specification probability, at month 36. So there's a better interpretability about that. So with that, we saw these two approaches are fairly similar for 6 batches. And now we can go over to the second case study to show where the real power of linear mixed effects models lie. Here we have 9 batches, so 3 batches more than before. However, we have a specialty. We have 3 dosage forms. One concentration comes to 3 batches, the other one with 5, and the third one with just 1 batch. Again, we have a lower and upper specification limits, but because we only have a tendency downwards, we only look at the lower specification. And again, as a theoretical aim, we want to extend the shelf life to 36 months. Here, the situation is like that the CQA, which we're looking at, has different levels for each concentration. That means they would need to be submitted for the extension separately. And thus, these data would not be submittable because we have one with only 1 batch. Because the ICH says, and also, of course, common sense says, you should not give information about a product with just 1 batch. However, from biochemistry and also from model selection, we know, or we can deduce, that all dosage forms share the same variability on their own level. With this, we came up with the idea to interconnect these data sets by implementing the concentrations as additional fixed effects. I don't want to go into detail, but here again, the 6 and the RIX models were chosen. With that, we come to the model definition. In the ICH approach, nothing changes. Like this model, we could kind of say we have different overall intercepts for each concentration. This would be a different parameterization, but it wouldn't change the model at all. The only thing which we get here by interconnecting these 3 data sets is that the measurement error, so the standard deviation of our measurement error, is estimated over all 9 batches, which is at first, on the first sight, a good thing. Also, what is different here, although this is a whole model, the shelf lives would be different for each concentration. However, in the Bayesian linear mixed effects model, the situation is a bit different. Here, we can apply a overall intercept for each concentration differently, because here the overall intercept is not one of the batches. It's really an overall for the variation, an overall for the concentration. And these deviations from that to get the realizations of the batch distribution are modeled such that we have for all concentrations only 1 standard deviation, because we could we assume, or we could show that the variability is shared across the different concentrations. So here again, we have population parameters, product parameters. Here 3, here 1 for the common slope, and here another one, so in total 5. And if we estimate these, we can again then get 3 batch distributions and get our shelf life as I discussed earlier. So here you see the results for the ICH approach. You can already see that this looks sparse, definitely, and of course, this looks very, very suspicious. However, it kind of induces that we should be able in all concentrations, if we would believe our model, that we can use 36 months as an extension shelf life. In the Bayesian linear mixed effect model, we also again get these realization estimation of the batches, which look similar as in the ICH approach. And also when you look at the shelf lives, you see they are fairly similar, some longer, some shorter. However, interesting gets when we, of course, look at the product shelf lives. Here, kind of the minimum of the batches for each concentration of the shelf lives, and here overall concentrations separately for each concentration for the batch distributions. And of course, we are fairly similar in the second concentration and the first concentration, although the Bayesian linear mixed effect model is a bit more conservative. But the biggest difference, of course, you see here again, where we would just slightly make it for 36 months in the Bayesian linear mixed effect model. However, in the ICH approach, we would be 9 months longer. And again, of course, we get here the OOS probabilities. Okay, so with this, we can see that if we do not include the batch-to-batch variability in the model, we can get here really bad results. However, if we can connect these data sets, we can kind of borrow us the information from other data sets to 1 for the variability, if this assumption is correct. And with that, I want to finish my talk. The 3 dots are for the points in the beginning. I don't want to re-summarize them again. The only thing I want to highlight again, the ICH approach is kind of just a minimum approach with a low number of observations. And of course, from a statistic point of view, this is not a good approach because there is no uncertainty estimation in there. The uncertainty comes from the number of batches here in the min-max in the minimum approach, and a 3 number of batches, 3 batches is not a good number. Here, the linear mixed effect model models and utilizes the collective batch behavior, which I said is the product behavior, is kind of what we are really interested in. However, for 6 batches, we saw that both approaches are fairly similar. Here, we could also see that the Bayesian approach adds some simplicity and interpretability to our approach, which is nice. And also, I didn't go into detail. We can go into detail in the discussion. The linear mixed effects model does not come with so much more assumptions which have to be tested, maximally 2 more. In this novel approach of interconnecting data sets, we saw that there is no benefit by interconnection in the ICH, yet it is even harmful. But of course, no one would ever do that. However, we could see that the linear mixed effect model is able to intuitively interconnect the data sets, and with that help in situations where we might have incomplete data sets and still being able to infer the batch uncertainty-based shelf lives for several dosage forms if we can make a connection between them. And with that, in this situation, the linear mixed effect model would have allowed us to quicker submit the extension while still retaining the patient's safety. With this, I would like to thank you for your attention, and I'm happy to discuss questions and get comments to that.
Thank you very much, Tobias. We're really good in time, so before the break, I would say we can take a couple of questions. I already see 2 in the chat, but Theo, you also turned on your camera, so please go ahead. And you don't want to put yourself first, I see. Okay. Then let's go with Jose's question first. Jose asked you, Tobias, can you talk about the choice of price for the fixed effects and variance components?
Okay, this is a very good question, because the fixed effect model is based on in a frequentist approach. Why did I do that? Because I wanted to be kind of true to the approaches which are done now, because the ICH usually is done in the frequentist way, and the new one is done in the Bayesian. I wanted to have this complete opposition. However, for the mixed linear model, I chose flat priors, just to be, let's say, we wanted to use flat priors because we had the feeling when we want to go into this comparison, we do not want to give any kind of candies at first to the Bayesian approach.
Thanks. But can you mention what flat priors do you use? Do you use an inverse gamma? What type of distribution do you use? Sorry for not being more specific.
Yeah, no problem. No, nothing at all. Completely flat.
Oh, okay.
And it works nicely.
All right, then one more question from Ben. You demonstrated a comparison between the ICH fixed batch model and the Bayesian model, but wouldn't it be more apples to apples to compare a random batch model to the Bayesian model?
I would say no. What I kind of definitely understand, it would be maybe kind of, we do two steps here. We do a step from fixed effect model to linear mixed effect model, and we do a step from frequentist to Bayesian. So it kind of would have been cool if we could have separated this. I completely agree there. However, we do not see how we could have done the testing in the polyvalidity tests in a hypothesis way, in a frequentist way, which would have satisfied us. And that's why we did kind of the double comparison, the double switch in both steps.
Yeah, sure, that makes sense. It is more of a challenge to compare, to do the model reduction testing in a random approach versus the fixed, which is outlined in the ICH. But there are ways to do that. But no, thanks for your presentation. Very interesting.
Oh, it would be interesting to hear about it. Maybe we can talk later.
Theo.
Yes, actually, I just want to say that Stan sent a question, but he sent it directly to me. And the question was, Tobias, what software did you use?
I did this in R. Both for the Bayesian approach, I used the Brooms package. I played around with the StanRack ARM, which I don't like that much. I used it for some years, but I got a fan of the Brooms one because there's a lot of in there. Because I didn't show regarding the model selection, I also did leave one out or leave 10% out and the stuff. Didn't show that here, and this is all implemented in Brooms package easily, which of course in the Bayesian framework takes some time, but you can start it
possible.
But
so
possible.
You said Brooms package in our...
Yeah, BRMS.
BRMS, okay. Yeah.
I'm not sure if it's called brooms. I call it brooms.
Sure.
Although
BRMS. Okay.
Yeah, I speak it brooms, let's say.
Right. Thank you.
And I see like if
there-
I'm some dark lord here. Yeah, my light is not working up there. That's why I only have the table lamp.
we'll still see some kind of silhouette, so it's fine.
I think there was one more question by John. Do you want to cover that, Timo?
Exactly. This was, I would now take this one, Tobias. One more question from John about whether you have already used this approach in submissions, whether it has been accepted by regulators, and if you included the interconnecting data sets with it.
Yes, no, these are case studies here where we use real data, but it was not used in submission. However, this approach we used just recently, it's not submitted yet. No, sorry, it is submitted, but sadly the regulators did not want to see the statistic at all. They were okay with just the data.
So it appears the data itself was convincing enough.
Definitely. But of course, I would have been lucky if they look at it.
So I think one more question by Jose in the chat. I think we can take this one then and head into the break. So the brooms of BRMS seem slow in some situations. Did you run into that?
Did you run into?
Did you run into that you find it to be very slow?
To be honest, no. I didn't realize a big difference to this rec arm one. But also, to be honest, these models are not really big. Like 6 batches or 9 batches with so less parameters. This is not a problem for the nuts in general, I would say. So let me think, because for model selection, of course, I ran through all models. Especially for the interconnecting data sets, I also checked if we have an interconnection between slope and intercept for the concentration and stuff. I think it was half an hour to do like 60 models or something. So it's okay.
Thanks.
I think there's a lot of good questions here. Maybe we should keep some for the panel discussion for later.
Yeah, really good question.
Absolutely. Yeah, let's wrap it up, the first part of today's session with that. So thank you again very much, Tobias. Also, thank you, Gang and Mark, for the presentation so far. Before we proceed to the parallel breakout sessions, as you used to over the last few days, we are having a break until, let me look, 20 past 11, so roughly 15 minutes. After the break, everybody is invited to come back to this main room. And at this point of time, the breakout session should already be available and you can just click whatever you want. Yeah, welcome back, everybody, after the breakout sessions. I'm sure you all had a lively and a good discussion. I mean, one breakout session I could join and the other one was Bayesian. So I'm pretty sure there was a good and a vivid discussion on that. So for the last case study of today, and therefore actually the last presentation of this year's workshop, I would like to invite Christian Oliva-Vies to the stage. Christian is going to talk about stability data quality from an Arrhenius perspective. Christian works as a principal statistical scientist at Genentech Roche and is currently a member of the non-clinical biostat team, where he offers statistical support to scientists and engineers within pharma technical development. So, Christian, the floor is yours.
Thank you, Timo. Let me first share my screen just to make sure that everything is okay. Okay, can you please confirm that you can all see my screen here?
Looks perfect.
Awesome. Well, so first of all, thank you, Timo and Theo, for your invitation to give this presentation on this conference. It is great to be here, and also thank you people for staying here until the last presentation of this year conference. So today I'm gonna be providing some insights on stability data quality from an Arrhenius perspective.
Did I disconnect or did Christian just disconnect? Now you're back, Christian, right?
You're back, Christian. Christian, do you want to turn off your camera, perhaps?
You were frozen for a little bit.
It looks like he's frozen again.
Yes.
Christian, now you're back.
Yes, he has some technical difficulties.
If it doesn't work, I can share the screen for you and then you can just tell me next slide if it's.
Yeah, can you hear me?
Yeah, we can hear you now, Christian.
Okay, I am sorry. We're having some interruptions because I am now in Florida and we had a hurricane in the morning. So maybe that's why the internet is failing. Okay, so if this happens again, please let me know and I will try to do my best.
All right.
Okay, so I was just saying that I'm going to be talking about stability data quality from an Arrhenius perspective. I'm providing some insights. I mentioned this is not a use case as the two previous presentations. It's just sharing some insights that we and some features and properties we need to look at when collecting stability data with the purpose of doing extrapolations. So the outline of the presentation is I'm going to start talking about Arrhenius kinetics, like why are we interested in Arrhenius kinetics these days? Then I'm going to move to Arrhenius-based models. So I'm going to be presenting just some Arrhenius-based models, at least some of the ones that I have seen in recent papers. I'm going to be just showing them just to give you an idea what are some of the parameters they have. And then I'm going to move to the main part, which is stability data quality. So I'm going to briefly give one definition of data quality, and then I'm going to talk about what are some features that we need to look in stability data so this can be considered good data quality. So let's start with the motivation. So the main motivation is the acceleration of biologics development. So as you are aware, in the pharma industry, there's an aspiration to accelerate product development of biologics. And one thing we need to keep in mind is that product quality cannot be compromised by that acceleration. Its regulatory agency allow for expedited approval of therapies through breakthrough designations. However, even when we have those expedited pathways, stability studies are in the critical path for this acceleration. The main reason is because ICH guidelines, such as the Q1A, require us to collect real-time long-term storage condition to determine shelf life. So when we start thinking about that is, well, if we want to have an initial commercial shelf life of 24 months, then according to these ICH guidelines, then we need to have at least 18 months or close to that. So we're already talking that in terms of the acceleration, we need to collect data for that long. And so in that case, as an alternative, we have the Arrhenius kinetics. So people and collaborators are looking efficient to generate stability data at accelerated conditions rather than the long-term storage condition. And then the question is, well, how can we use this accelerated data that we're generating to say something about shelf life, right? How can we use data, I don't know, maybe 20, 25C, 30C to say something about what is happening at the 5C condition? So in that case, we have the Arrhenius equation, which links the temperature dependence of reaction rates. So that is great, right? It's like we have an equation that is linking temperature dependence of reaction rates. So if we have an equation, then as statisticians, we can say, well, we're ready to make predictions at the long-term condition, right? And you can see here in the right side, we have like a plot. I am just showing So the question is, can we make predictions now? And well, not so fast. And the main reason is because, as George Box mentioned, all models are wrong, but some of them are useful. And we need to make sure if this model or models that are similar to, let's say, to the baseline Arrhenius equation are in fact useful for our cases. So let's talk a little about some of the Arrhenius. Oh. Can you hear me?
Yes.
Yes.
Okay. Okay. I got a message that my internet was unstable. Thank you. So now I'm gonna talk about some Arrhenius-based models. And as a disclaimer, I would like to say that here I am only presenting these models just to have some background on what are some of the parameters that are included in these models. I am not presenting these models as a way of endorsing their validity for shelf life determination. I think that is really up to the specific use case we have, depending on whether we have a small molecule or a biologic, or whether we are studying one quality attribute or another. So this is just to get some background on what are some of the parameters we usually find in these models. So let's just start with the baseline Arrhenius equation. So as you all know, we have these 5 different pieces in the Arrhenius equation. We have K to be the rate constant. Then we have the temperature, usually in Kelvin. And then we have these 2, I'm gonna call them parameters, which are the frequency factor A and the activation energy EA, and finally a constant, which is the gas constant R. And here we have 2 plots. They are equivalent. The plot to the left is just a plot based on this form of the Arrhenius equation. The plot in the right side is usually more known. And the main reason is because once you take the log in the 2 places of the equation, then you get a linear model where your input is 1/t and your output is the log of the rate constant. So basically, if you can just impose a very simple linear model in there, then this is definitely more friendly to use. The important thing to keep in mind here is that this baseline Arrhenius equation is based on basic chemical reactions. So if we are studying more complex or heterogeneous reacting species, they might not be appropriately described by this baseline Arrhenius equation. So as an alternative, or one of different alternatives, we have this modified Arrhenius equation where we have, or we added a humidity factor. This is mainly used for small molecules. As you know, for small molecules, humidity is one parameter that affects the degradation. And as you can see, the form, it looks very, very similar to the, let's say, the transform Arrhenius equation. The only difference is that now we have this additional set of parameters, or parameter B, which is the humidity sensitivity factor, and then that is a parameter for the percent relative humidity. The interesting piece in this equation is that you can see that temperature and humidity, they are independent. There's no cross term for the 2 of them, which means that if you have a fixed relative humidity, then a linear Arrhenius behavior is observed, and we can see that in this plot. So in this specific example, we have they are fixing 60% relative humidity, and based on that, they are measuring 6 different conditions, and they are showing that there is a linear relationship. But as they actually change the percentage of relative humidity, then this is actually not aligned with the rest. So this is just one modification that is that has been used for small molecules. Then we have a second modification that is a bit more complex. Now we are facing or working with differential equations. They usually call this advanced kinetics modeling. So here I have an example where we have one of these kinetic models for 2 reaction stages. You can actually have multiple of them, like 3, 4, or you can have even only one. And though they will be under the same family of models. So this is basically saying like, what is the change, the rate of change over time of the attribute we're observing, and according to this differential equation, it is following this form. So in here, what we have? Well, we have alpha first to be the reaction extent. And we have the 1 minus alpha and the alpha. We have both of them, and then we have the different reaction orders, N1, M1, and N2, M2. And then we have also the reaction rate. So we have K1 to be one reaction rate and K2 to be the second reaction rate. And here, I will say that the notation used in this specific equation is poor, because K1 is not just one specific value, but it's actually a value that's going to change with temperature. So if you have, as in this case, if you have 5 different temperatures, then K1 is actually going to take 5 different values, and K2 is going to take another 5 different values. In practice, these reaction rates, both K1 and K2, they are modeled using the baseline Arrhenius equation. So in this specific example, with 2 reaction stages, if we consider the N and the M's and also the K1 and K2 to be modeled as an Arrhenius equation, then we are going to have 8 parameters. We're going to basically have 2 parameters for N, 2 parameters for M, and then each K1 and K2 are going to have 2 parameters each. So each of them is going to have its own activation energy and its own what was the other, the A parameter. So we're going to have a total of 8 different parameters just to explain this differential equation, or this set of differential equations. N and M, they are sometimes treated as fixed constants, or sometimes they are treated as parameters. So in the example I was showing before, I was saying, well, if we consider N and M to be parameters, then we end up being 8 or having 8 different parameters. And those parameters, they don't even need to be integers. So the first thing we might think is, well, maybe this is going to be 1 and 2, or 1 and 0, or maybe values between 0, 1, and 2, but they actually don't have to. They can be like 0.5, a third, or 1.25. Sometimes in some papers, what they do is they try to fix them based on what is expected. So they try to take into account the information we have about the reactions that are expected, and that depends on the quality attribute. And they try to narrow these values to be, let's say, if you're expecting just the 1 minus alpha trend to appear, but not the alpha, you might make the M1 to be 0 and the M2 to be 0 as well, if you have some information. So this is usually treated in one way or another. And there might be more Arrhenius-based models to consider. The important message I want to send by showing these 3 different Arrhenius-based models is that as the complexity of these models increases, the number of parameters that we need to estimate also increase. So I mentioned before, we have 2 reaction rates in that last model that I presented. We have 8 parameters, but we actually include a 3rd reaction rate. It definitely increases. And as we increase more the number of reaction rates, then the number of parameters definitely blows up. So given that we want to learn those parameters, then we need to use or collect the appropriate stability data so that each of the different features depicted by the model parameters can be learned. And this is not really about how much data we have or how much data we collect, but about the data quality we are collecting. From that point of view, data quality is not only about capturing data correctly. So here I have the very simple example of having rounded data versus unrounded. I am not only referring to that when I'm saying data quality. I'm also talking about, is the study design appropriate to answer the question of interest, or are we studying enough time points, enough temperatures, or enough lots? We also need to think about that when thinking of data quality. And just remember that the fitted model can only be as good as the quality of the data used to build. So if we don't have good data quality, then we're going to be feeding a model that maybe is not going to be useful for the purpose of getting a reasonable prediction of the reaction rate at the long-term condition. So with that, I'm going to move now to stability data quality. And here I have a very naive definition. I actually found this definition just searching on the internet. And it says the ability of a data set to serve an intended purpose. I think this is similar to what I was explaining before. It's not just about rounded versus unrounded, but it's also about, do we have the data we need to make a reasonable prediction in this specific case? Here I have something a bit more official, let's say, like I have here a quote from a paper in 1999. It was a roundtable on research and development of biologics that says, in the past 10 to 20 years in the US, the quality of data from clinical trials has improved remarkably. Historically, most data problems have been the result of poorly trained investigators who either enrolled the wrong patients, did not follow the clinical trial protocol, did not record the required information, or had poorly designed protocols. So even when here they are talking about clinical trials, they are in general mentioning that even designing a poor protocol is not going to be helpful for data quality. And that is part of what I'm going to talk in this section. When thinking on stability data quality with the purpose of using the Arrhenius or an Arrhenius-based models to make a prediction, there are two main features that we need to consider or we need to learn. The first one is the degradation over time per each condition. So if we have, let's say, 25, 30, 40 C conditions, then we need to look at the degradation rate for each of those different conditions. And here the main question will be, are we observing a linear trend or are we actually observing a nonlinear trend? If it's nonlinear, do we have enough data to actually know the nonlinearity? Is the nonlinearity going to be exponential, logarithmic, is it going to be quadratic? What do we have to actually impose something in there? The second part, the second relationship that we need to learn is the temperature dependence of reaction rates, right? And here, the main question is, well, we have that baseline Arrhenius equation, and when you take the log of that Arrhenius equation, you get like what I'm calling a linear Arrhenius behavior. But the question is, are rates expected to follow the linear Arrhenius behavior, especially for biologics? It's like if we take high molecular waveforms and we have the rates at different conditions and we plot them using this Arrhenius plot or linear Arrhenius plot, are we in fact gonna confirm the linearity that we are assuming? So these two features are something that we need to capture in the data. So we need the data to be loud and clear regarding these two properties. That is, the data needs to show clear evidence of any of these relationships that we are assuming. And that is very important, especially because we are going to be doing extrapolation. An appropriate design, of course, for stability studies will help to collect data of good quality. And now here I have some examples, more illustrative examples, just to make some points. So for instance, in here, we have, we're looking first at the first feature, right? What is the degradation trend over time? So let's suppose we have 25C data. We're studying 25C data from 0 to 6 months. And the true degradation trend is something like this. So it's actually not linear, but we observe some curvature in there. But then in our study, we just do a study from 0 to 3 months, and we sample every month. If we do that, given that we are not going, we have a short study, we're not going all the way up to 6 months, then based on the data we're going to be collecting, maybe we're just going to see that linearity is good enough. And in fact, if we were to try to put here some non-linearity, it's going to be hard because the data is not showing that. So we're just going to be making up any curvature we want, but not actually getting that from the data. So this is one example where you are studying something, but not enough. And I would like to say that in most cases where we study these accelerated studies of 25C, for instance, it's because sometimes we want to perform a total change calculation. We want to know, like, if we have like different steps and one of those steps is having a 25C, we want to grab a small budget, and that's the reason we're estimating that specific rate. And sometimes going all the way to 3 months is more than enough, because we're not expecting to put drug product at 3 months for that the 25C condition. Maybe we're going to be putting that at 15 days, a month. So 3 months should be enough. But in terms of learning the trend, so we can inform the Arrhenius model, we actually need to have longer studies. As a second example, again, talking about the degradation trend over time, it might be the case that you in fact go all the way up to 6 months, but then you have like some gaps in there. So here we have the 0 time point, the 1 month time point, the 3 months, and then you go to the 6 months. Then here is like, well, something is happening, right? It's like when you look at that, like, well, yeah, definitely something is happening at 6 months, but it is not very well captured because there is a big gap from 3 to 6 months. And when you see that, one question that we might ask is, well, are we getting this value to be high just due to assay variability? Or maybe even more, is it because the fact that we're testing in a different session is making this value to be higher than it should be? So here's when you start like not having enough evidence to say, do we have linearity or should we try to use some non-linearity to capture that? We don't have enough in the data. But then if we actually have, as in this example, like every month, then we're going to have more evidence that there is some curvature, and now the data is going to allow me to capture that trend without having to make to make up assumptions on how do I believe the curvature is going to happen within these 6 months. Important to mention is that the curvature that I'm showing here for 25C happening probably somewhere between 3 months to 6 months. It might be observed in earlier time points as the temperatures go higher. So if we were to have 40C, maybe the curvature, if any, is going to happen like getting close to the first or the second month. And maybe for the 5C condition, we're actually going to see curvature once we get to 60 months or something like that. So depending on where do we believe this curvature might happen, we might need to sample more time points so we actually capture those trends. Now, let's move to the second property. Now we're talking about the temperature dependence of reaction rates. So reaction rates need to be sufficient. Sorry, the temperatures we study need to be sufficient so that reaction rates can be learned, or the relationship between the reaction rates can be learned. So here I am starting with a very, very naive example. So let's say you only have 2 different temperatures. You have the 25C temperature and the 30C temperatures, and here you have the 2 reaction rates that correspond to that. So the question is, again, can you fit an Arrhenius model? And here I am thinking, can you fit the baseline Arrhenius model? So yeah, of course, it's like, as you noted, we are just gonna fit a line, and if we have 2 points, that's the minimum we need to fit a line. So yeah, sure, we can do it. There you have. Here I have the corresponding fitted model, and here where we have the cross between the red dotted line and the blue line, here we're going to have the prediction of 5C based on these. But then the question is, given that you only have 2 temperatures, is like, is that 5C prediction scientifically meaningful? And here is when we start having those questions about mathematically we can provide an estimate or a prediction, but the question is there a scientific meaning to that specific prediction that we're making? So let's move now to, well, let's say, okay, 2 is the minimum. Let's take 3. Let's add the 40C. Now we have 3 conditions. Can you make a prediction now? It's like, well, of course, if I was able to do it for 2, I can do it for 3 again. I can make a prediction in there. But then the question is similar as before, is, well, even when we're using these 3 different temperatures to make a prediction of 5C, the question again is the trend expected to be linear close to the region where we desire to make a prediction? That we don't know because we don't have any evidence to say something about that. And the most important piece is beware of the distant extrapolation we're making. So here we have the 25C, which is the closest to the 5C value that we want to predict. So you can see that this is actually farther than we would like. And in general, we know that we need to be careful when making extrapolations. In this specific example, you can see that the true relationship that I used to generate those 3 time points is in fact a convex relationship. So the reaction rates are not actually following a linear relationship or a linear Arrhenius relationship. They are in fact following something more convex. And this might happen actually for heterogeneous species. They are not expected to to follow the simplicity of the linear Arrhenius model. So in that case, what is happening is that we are in fact getting a prediction that underestimates the reaction rate. And if we believe that we are going to be setting or determining shelf life based on the prediction we're getting, then that is going to be not the best scenario, right? Because we are actually underestimating, so we're going to get a longer shelf life than the actual shelf life that we should have. So what to do in that case? Well, adding temperatures that are closer to that prediction of interest might help. So for instance, in here, we have, I am here including 6 temperatures, so from 15 to 40. So every 5, I am including 1 temperature. I'm getting these 6. These are now showing a bit of curvature. I won't say this is like definitely a very clear and evident curvature happening. It might not be the case, but at least just having these time points, especially the ones that are closer to 5C, they might provide some evidence that if we try to fit a linear relationship, we're going to be conservative, and then we're going to have to do something else. Important also to mention is that, and this is also related to data quality, is that in this specific case where we see that the curvature is not that evident, but at least we have some evidence that this is not a linear relationship. If we have high variability in each of these reaction rates, then we're not even going to be able to capture any curvature, which is a good call for generating and better assays that are less variable, so we can actually learn these trends more appropriately. So finally, as a summary, as I mentioned, these Arrhenius-based models have been considered as a risk-based modeling strategy for shelf life prediction of biologics. And it's important to consider the quality of the stability data, but from an Arrhenius perspective. The stability study designs should be based on specific durations of the studies, time points, and temperatures. Again, thinking on what is the information we need so we actually inform the parameters by the data and not just give or produce predictions that are completely based because of the model. And of course, that the good quality of stability data comes with an additional price. So we might need to generate longer studies, we might need to do more testing because of more time points, and we might need to have better assays. The models need to follow the science and not the other way around. So as I mentioned, given the long extrapolation that is required, the underlying properties of the assumed model might cause bias in predictions. So sometimes if we are assuming, let's say, that there's going to be an asymptote, but the data is not showing that asymptote, then the model is going to drive the asymptote, but not the data. And that might give us a biased prediction. And finally, and in practice, we have in practice that we always appreciate having good accuracy in our predictions, but in this specific case, I think having a conservative shelf life prediction might be of great value, as long as the conservativeness of that prediction can be defended. So what I'm trying to say here is that I don't think, it's not like we're going to get a prize for getting the best prediction or the best accuracy in our predictions, but as long as we can actually come up with something that helps us to have a reasonable prediction, even when it's not accurate, but it's conservative, so that we can determine shelf life at 5C, that might be very helpful for the purpose of shelf life determination. And with that, I'm going to thank you all for being here. I'm going to be open for any questions you might have.
Thank you very much, Christian. Thank you for this wonderful talk. And Jose, I see you already raised your hand. I would say we take one more question before then heading to the panel discussion. But Jose, go ahead.
Well, I can save it for the panel discussion because I mean,
just go ahead. That's fine.
Okay. Well, Christian, thank you so much. Really, two things that I really like seeing. So one is the use of Arrhenius models in this context. I came from semiconductors and material science, so I did a lot of work with these type of models, setting up shelf life, also using acceleration factors between temperatures to evaluate, how the life of a material will change. When I got to biotech and I started looking at some data and I look at stability data, I thought, oh, this is, the realm of Arrhenius. But I was told by some of the statisticians and people doing statistics there that no, the Arrhenius does not apply to biological phenomena. So that's one question for you and then for the panel to discuss. And the other thing I really like is you talking about the design of the stability studies, because one of the things I always thought that is really poor is how we do stability with so few data points over time and all that. So when you do these studies that you pointed out, it's not only how many time points, but the number of time points will change depending on the temperature and all of that. So it's a big DOE principles that come into play. That's all. Thanks.
Thank you, Jose, for your comments.
All right. Then I would say let's immediately jump to the panel discussion. Christian, your connection now at the end worked perfectly. I think if you now stop sharing, it might be worth it to try to turn on the camera, but if you feel more comfortable with the internet connection, you can also stay without your camera. But nevertheless, I would also like to invite the other presenters of today to come back online, to come back and turn on the cameras and join me in the panel discussion. Gang, I think Mark already left. I didn't see him in the audience. Is this right?
Yeah, look like so.
Okay, all right. So perfect. Tobias, Gang, Theo, and Christian are here. So yeah, thank you everybody for the presentations. You know the drill in the panel discussions. Just start asking a question, and maybe just to get it started, I would point out one question to all the three of you now. So you all provided case studies or ideas of new or advanced CMC statistics methodologies, and if you recap on the last 3 days and where we had different discussions, especially considering yesterday's session about regulatory advancements, what do you think about the acceptance and or applicability of these methodologies, being it with respect to different stakeholders such as your team, your organization, or also from a regulatory body? Whoever wants to go first, I would be really keen in getting your thoughts on that.
Tim, I can probably start, given that I was the last one to talk. I can probably start. So Timo, I think so the first thing I would like to mention is that the use of these Arrhenius-based models to make predictions. At this point, ICH guidelines do not really allow for the use of these models, at least not openly. But the good thing is that ICH is going through a revision or will be going through a revision that might or might not open the window for starting using some of these models. However, what I do think is that we need to show, like, it doesn't matter which of these models we're using or a different one. We need to show, based on data, that the models that we're using, they are producing predictions that are useful for the purpose of determining shelf life. And what I'm saying that is because I have seen internally and externally cases where we try to make predictions where we have where we are using stability studies that were not designed to say something from an Arrhenius perspective. As the comment from Jose before, it's like we have a limited number of time points, or we have even a limited number of temperatures, maybe 2 or 3. Sometimes we don't even know if we are considering temperatures that are above certain threshold that might actually inform the prediction at 5C. So let's say maybe we're trying to use like accelerated data at 60, 70, and 80 degrees to say something about the 5C, but you don't know if that trend is going to be actually applicable. So I think we do need to develop, to produce more data first, So we can actually check the models and see what kind of predictions we're getting and how useful they are. Because I think the use cases I have seen so far, internally and externally, are very limited in terms of the data that is used to fit the different models.
Thank you.
I could go next about my thoughts on that. So I think my answer will be twofold. I think first part is many times this new methodology is kind of necessary to solve the problem we have. For example, many times we have to use Bayesian or the methodology I talked about in my presentation. It's just we don't really have a good way of using traditional method to do it. So we many times and the business needs push us to use new methodologies. And another part of my answer is really many of new methodology can give us predictions on new products we are going to launch. Many times, for example, Arrhenius relationship, and we find there are a really interesting universality across our products regarding Arrhenius behavior. So we can prove that is really the case. And in the future, we can do fewer stability studies if that is holds for all of our products. So that is a huge value to our stakeholders and collaborators. And I kind of feel if we can build a model that have really good predictive power, then that is a really easy sell to our company. So that's my thoughts.
Thank you, Gang.
Yeah, I try to go last because I'm still not sure what to answer exactly. Like Tara had nice comments in the breakout room, Bayesian. She said that they already submitted several Bayesian methods, Bayesian use cases, and never really got a pushback. So what she also said, maybe we are just too scared oftentimes to just try it and use it, especially when we see that here, for example, in this case, Bayesian is a benefit to this situation, or where frequentist approaches would be kind of bent to answer questions which it's not made for. So in that, maybe we need to get more bold to just submit it. But of course, we as a statisticians, we are not the one to decide that usually. So I think there are some, it would be really interesting to have direct answers over that from regulatory regulators directly. That would be really interesting to go into one-on-one discussions with them.
Thank you all. So I have a couple of questions in the chat, but Julia, you raised your hand pretty early, and that's why I would like to give the mic to you.
Oh, sure. Thank you. Yeah, these were all excellent presentations. Thank you all for the work you put into them. I would say I have a question on for the last presenter about the Arrhenius stability. Really enjoyed that, and I find it very relevant. I'm wondering if you have considered using a model-based design approach to address the issue of, I like the way you put it, data quality for stability studies, and deciding where to fill the gap. So we have a very similar issue, probably many of you do, where we have the data that we had to collect for standard stability studies. We actually have an extremely high volume of data now, but we also are trying to build a predictive mechanistically based model. And so we're, one of the things we're considering is maybe model-based design, looking at what would be the most informative points to add to our collection. So just wondering if you've tried that.
Julia, thank you for your comment. I actually so we did, and we actually encountered a similar issue to what you were describing. We have a lot of data. So in terms of quantity, we have a lot of historical data that we have produced. But when we were actually looking at that data is when we started realizing that some of the properties that were expected, they were not actually appearing in the data. So we were thinking, what is happening here? And the first hypothesis was that, well, it looks like we are not studying maybe long enough. Like I can bring an example that is like at some point we were looking at high molecular weight forms. No, was it? Yeah, I think it was, sorry, it was acidics. We were looking at acidics. And then we were thinking, well, maybe there's a first order reaction happening there. And then we were looking at the accelerated data that we had, and we didn't see anything. It was like completely flat, or not flat, but it was a straight line. So it's like, how can I fit or try to fit a model that considers a first order reaction? The first order reaction is just basically going to try to push everything to be a straight line. So yes, we did observe that, and part of this presentation was, or the motivation for presenting this topic was specifically what you said. It's like when we started looking at the different models, or let's say these three models that I just presented, we started looking at what are some of the pieces or some of the parameters and what they are explaining. Like for instance, in those advanced kinetic modeling models, they have the potential S shape. So we were thinking, if we want to fit something like that, then we need to have data that actually shows that the asymptote is actually appearing. And we were looking at the data we have, and we were like, well, that is not clear in most of the times. So we need to do something else. So I think what you said is a good summary of what I tried, the message I'm trying to send today. It's like, depending on what the models need, you need to look at the data and need to make sure that you're going to be explaining that. And you definitely, I think you can definitely use historical data to inform also. Like for instance, if whether or not you're expecting to have some curvature in there happening, and if so, you can maybe use that historical data to inform your study design and say, well, maybe I need to locate more time points within this time frame, or not. So I think Julia, what you said was the main reason for making this specific presentation.
Perfect. Tim, I would go next to you.
Yeah, my question was very similar to Julia's, and it really brings the several talks together. Because again, we had differential equations in the one talk, which were the mechanistic understanding of what was going on. And I've come to, realize that certainly science trumps everything. It trumps the models that we fit. And to the point that was just made, it's possible that the models we fit don't meet our assumptions. I would think that's a rather precarious situation. Modeling data that don't meet our mechanistic assumptions is something I think we really need to figure out. As an example, for the last presentation, when you were presenting your times for your expected kinetics, you had first order kinetics. You were in a linear scale, and if you'd taken the log of the vertical axis, you would have had a straight line. So I'm not quite sure what you meant. You always have a straight line. You were describing that as that was not desirable. But I think Arrhenius requires that you have a slope, not that you have curvature, not that you have an S shape. So I'm not sure how you even enter into Arrhenius if you can't understand the mechanism that generates the order of kinetics that you're trying to model. So the bottom line is I think we'll have everybody, or statisticians should have great success if they can back up their modeling with mechanistic understanding, not just empirical fitting, but mechanistic understanding. That'll put you a lot closer to, I think, acceptance, maybe even the answer to Jose's question. That is, if you know mechanistically what's going on, then the old dogma that you can't apply this to biologics and or vaccines should go away. you're actually describing what you know to be happening to the system.
Yeah, this also addresses one or the other question from the chat. Yeah, thank you, Tim, for that. Theo, you want to go next?
Yeah, I just want to go back to that first question you had, Timo, to the whole panel about acceptance. And this question is for Tobias. I really liked your presentation. I liked your slides, with showing how the ICH guideline basically makes us do all this snakes and ladders kind of procedure, go here and here. And I really liked your comment about, the null model and how we're misapplying hypothesis testing when we're doing this. But yeah, historically in our company, long time ago, another statistician said, "Oh, why are we doing this? Let's do mixed modeling from the get-go. Just a mixed model with random slope, random intercept. This covers all the bases that the ACH guideline actually proposes and tries to, fix with all the snakes and ladders. And we don't need to do snakes and ladders anymore. Just fit one model, linear mixed model." And that person tried to do that. And they were kind of successful, but not really, because of internal pressure to conform with this ICH guideline. They said, "Oh, it's not ICH compliant," but it was. So have you dealt with anything like that? How do we, it's not really just, the regulators, but sometimes internally there is this risk aversion. So I wonder if you can comment on that.
Timo is my boss, so I have to be careful. No, but from a colleague of mine, it was really interesting. He kind of, I helped him to apply it. So it was not my project where it was applied first. He was pretty skeptical in the beginning, because he's also not, let's say, fluent in Bayesian. So it was kind of a double jump for him. And not only the switch from fixed to mixed model, but also from frequentist to Bayesian. So there was a bit much in the beginning, I would say. But to be honest, after some discussions, I realized he was eager to understand. And at the end, I also could kind of convince him, and he also now is really interested and calls me several times to just talk about it. So yes, there is a starting skepticism, but if you have the time to talk, and of course he's a statistician, he can grasp the stuff, and I think it's possible to overcome this. Yes, and from Timo, there was, of course, in the beginning also some reluctance, but I think this is also, for example, like, although I like Bayesian, and I think we can do a lot of it, I think it's also kind of good that you are a bit reluctant in the beginning. So you should at first question it, because frequentism is kind of, this is the gold standard, it is used a lot, everyone knows it. So something new comes. So yes, of course, be skeptical at first, but be open to discuss it. And from there, I got a big support in our team, from at first skeptical support, but I got the support to try this.
Great, thank you.
So Tobias, maybe just I would point one question from the chat from Stan directly to you, and it's about the interconnected model. Stan says that in your interconnected model, I have found that the residual errors can be related to the strength or concentration of your product. Did you consider the possibility of heterogeneity in your model?
At first, I liked the name Stan because of Stan probabilistic language. But can you elaborate on the question? I think I'm not getting it 100%.
Maybe.
Yeah. So you mentioned in your interconnected model that you were including 3 concentrations, right? Yeah. Okay. So I'm just saying, in my experience, I found that the residual term. See, I'm assuming. I think you assumed a common error term across the concentrations, right?
Are you talking about the measurement error or the variation, the measurement error?
Residual error. Right.
Yes, correct.
Yeah.
This is a really good question. We did not made a model selection for that. But we kind of went with it, but it's definitely a good point of criticism. We went with it because the ICH also does it. So, but this is kind of, I'm biting my own tail because we wanted to criticize the ICH and make it better. So we could have gone the whole way, but we didn't take this into consideration. That's a really good point.
Okay, thanks.
So we also assume homoscedasticity over concentration, kind of, yes.
So maybe I go on with one question from Stan directly to Gang about which software you use for solving your PDEs.
Oh, that's a quick answer. Is MATLAB. You can kind of easily implement a solver in MATLAB to do that. Well, you can do that in any language, really. It's either Python or something like that. Yeah. So
I thought it would be Matt, but I just wanted to confirm that.
Yeah.
Yeah, that's I think mostly the standard answer you get when solving for PDEs. So I have three raised hands, and Tobias, you also raised your hand.
Yes, is that allowed to ask a speaker from a speaker?
Just speak up. You don't need to raise your hand. Just speak up.
Okay, yeah, I had a question for you, Christian. I also really liked your talk, and I was curious about these 2 easy plots where you started with 2 points and then went on to 3 points. This Arrhenius equation, the linearized one, in the log scale. Is it possible? Oh, no, it is possible, but was it done to infer like an error bar for your predictions? It should be something like this, I guess, that you can kind of then see the further you extrapolate how wide the uncertainty bands go. Like, was this done before?
Yes, that's a great question, Tobias. I actually did. I did that in the past, and you know, in this case, I mean, of course, I just said, like, as you get your prediction farther away from your data, this starts like opening up like a cone. But given that the reaction rates, at least the ones I built in a previous use case, are based on the average rate or the common slope, let's say, of combining different lots, then the uncertainty around the reaction rates were not that big, because it's like you're talking about the uncertainty of the averages or the average slope. So it does open up, but it actually doesn't open up that bad. If you're only using one lot, if you only have one lot, and you're just computing the reaction rate based on one lot, so one lot for basically creating that, you might have like a different story, because then the uncertainty that comes with that is going to be large. But in the transformation, it's not. However, one thing to notice is that there was this one plot where I showed like I fitted a curve. It wasn't just any curve I was able to fit, and you have like you're over predicting the actual or the true 5Z rate, right? Even when those 2 points look closer, when you go back to the untransformed scale, you get actually values that are different. It's like, because you're working on a different scale, small differences might be actually much bigger than you think they are. So what I'm trying to say, that even when you don't see these prediction bands opening up that bad, they are actually, when you go back to the untransformed scale, you can get actually very wide confidence intervals.
Yeah. Thank you.
Yeah, thank you too. Yes.
Theo.
Yes. I just want to clarify something first. What you said, Christian. Are you getting your uncertainty intervals by fitting the Arrhenius model, right? And not by fitting a line, right? You're fitting actually using all the data and then fitting the dynamic Arrhenius model and then getting uncertainty estimates through that, and not just by computing rates and then fitting a line, right?
That is correct, Theo. That is correct. And you know, Theo, that is a great point now that you mentioned that, because what I have seen that sometimes happen in practice is, you know, as I mentioned, is once you transform, you're working now in the log scale versus the 1/t. Sometimes what I have seen is that once you have the rates, then you fit the linear model. But when you do that, it's like you're kind of not really thinking on what are the assumptions that are coming with that, because the assumption, or the regular assumption, is that there's homoscedasticity happening, right? But that might not be true. So in most cases, we actually consider that when you fit the original model, not the transform model, but the original model, is like you have the exponential model plus your error, and that error in most cases is assumed to be mainly because of assay variability, and assay variability is considered to be, let's say, constant. It doesn't matter where you're testing, but when you are actually imposing the model in the transform, so based on the rates, and you still consider homoscedasticity, you're actually not following the assumptions, and you might get a great prediction that is not really capturing the let's say the noise or the variability that you should be capturing from your errors. Yep.
And I want to point out also that those uncertainty estimates, they are assuming that the Arrhenius model is correct, so it doesn't really give you an indication of how bad your prediction is if the Arrhenius model is not appropriate in for a certain molecule, for example, right? Or an attribute even. Yeah. So.
Yep. And that is so. I know as a statistician we do know, but sometimes when we are communicating with non-statisticians, we also need to be careful that the fact, so because sometimes you see these predictions, you have the prediction bands at the 5C condition, and the prediction band, they cover the real data. Right. And sometimes we believe that because the prediction bands are covering the real data, then our predictions are good. But I don't think that has to be true, because if you have like a terrible assay and you have like huge prediction bands, they are just going to cover basically anything you have. But that is not saying that your prediction is actually good. It really works with the trend. It's like one thing is capturing the trend, the other thing is capturing the error variability on the model.
Thank you.
Thank you,
Christian. You also raised your hand, right?
Yes, I actually have. I wanted to ask a question to Tobias, if that's okay. So Tobias, when I was looking at your the diagram, right, when you are showing, you start with random intercept, random slopes, and then you have like this testing. In your case, it was based on base factor, and then you have like other two decisions. The one thing I've been like thinking to myself is, it doesn't matter if you're doing a Bayesian approach or a frequentist approach. The fact that you have testing in the middle to determine the model is, I don't know, I find that to be a bit tricky, you know, because as you mentioned, we have the 0.25, and not only that, we actually, I mean, based on ICH Q1E, we're using this ANCOVA, which is not an equivalence step, but anyway, we're using it. So I guess what I wanted to ask you is, did you consider only having a random intercept, random slopes model, like all the time, like not doing any testing and just going with that?
Yeah, very good question. Because if you could say I didn't show it, but I had it in the backup slides. If you took a look at the random intercept, random slope model, it has a covariance matrix in there where it has these two variances for the intercept and for the slope. So if you just drop one out, make it 0, if the data says it is 0, then the matrix reduces to just a scalar, and then you have the same as separate as common slope. However, well, let's say I don't have a clear opinion to that, because the thing, the advantage for the RIAS model, you can also model dependencies. You can also model the correlation coefficient between slope and the intercept, which is really helpful. But this, of course, adds more parameters to estimate. Right. And what you see, the uncertainty bands, they make more like this trumpet thing. So I would say if you have, and this is what my example showed, if you have clear evidence, then why not go with the more favorable model? But for example, which is a really good thing, I did the model selection, but for example, I think this is what I take out from your nice comment. I did not, although inferred all models, I did not check the confidence bands or the credibility bands. So that would be interesting if it even makes a huge difference. And if you can always stay with the whole model, that's a good point. Yeah.
Yeah. Thank you,
Tim.
I'm still stuck on Arrhenius. And it's from a long history of trying to come to grips with it. And in a sense, going back to Jose's position, in the early days when I was considering Arrhenius, or at least learning about it, not considering it, and I was in vaccines where, again, if monoclonals were not modelable by Arrhenius, vaccines certainly weren't. But in any case, I was taught that the two-step fit, which ends up being the natural log of the absolute slope over 1 over absolute temperature, has a property to it that the points closer to the condition you're trying to predict, say 2 to 8, the slopes themselves have heterogeneous variability. That is, as you get closer to a shallow slope, the more shallow the slope, the higher the variability, which led me to conclude that the uncertainty blew up so much using Arrhenius that it was useless, even in the best of situations. And I hear you saying something that confirms that, but you're still advocating for it, and that is that the prediction intervals are very wide, is what you keep saying, and I'm not sure prediction intervals of what, but if they are the prediction intervals at the, say, planned storage condition, I'm not sure to the extent that we might use those limits on those intervals to set shelf life, that they're going to be very useful. And I want you to comment on that. And another fact, it concentrates on the degradation rate. In a traditional way of determining shelf life, there's also the level of the lots, that is the, let's say, alpha parameter. How do you account for that in translating Arrhenius prediction into shelf life determination a la conventional ICH guidelines, not doing the ICH guidelines, but the spirit of including the levels of the lots in the calculation?
Yep. Thank you for the two questions, Tim. I don't think that I have a good answer for that, and the main reason is because we are still starting to work on that. So, as the way I see this is, I see this, from the statistical point of view, I see the use of Arrhenius as a two-step process. The first one is trying to how to say it, like try to capture a reasonable trend, and the second step is like once we have a trend that is reasonable to be captured, then how do you measure the uncertainty? I think your first question was more about uncertainty. To be honest, we haven't gotten that far because so far the trends that we have been, the trends that we have been capturing with the data we have, they are not very encouraging. So the fact that they are not very encouraging is, at least for me, it's like I haven't been like focusing on gathering or getting the uncertainty, because at the end of the day, it's going to be not useful. It's going to be useless. So I haven't really take a deep dive on these parts of uncertainty, and I would like to add another layer of complexity when you start thinking on that. And I think you were asking that question. You were saying a prediction for what? And that is a good question for me, because let's say that you happen to have 3 lots where you are actually performing your stability studies at all of the different conditions that you need, like 6, 7 different storage conditions, accelerated conditions, not storage, 6, 7 storage conditions. So you're going to grab them, you're going to have 3 lots, and then you're going to build a prediction for what? Do you want to build a prediction for the average trend? You can do that, maybe, like you can compute the average or the common slope for at each temperature and use that to inform your prediction, and then you're going to have a prediction of the average trend. And then the question is, do we want to have a prediction of the average trend at 5C? Is an average what we need to establish shelf life? And if the question is no, then maybe we want a prediction of a specific lot, which is then more complex, because then you're requiring to have one lot on the same lot studied at all the different conditions. So I think that is putting another layer of complexity. And then the question about what do we want to get? Do we want a confidence interval? Do we want a prediction interval? Do we want a tolerance interval? That part, honestly, is still not clear in my mind. Like, what is exactly what uncertainty interval we want to compute in order to determine shelf life? And yeah, so I guess that's what I have to say about your first question.
Well, I think that last point you made is unambiguous. It's a confidence interval. That is, if we follow at least the spirit of the ICH guidelines, which uses confidence intervals, we're not trying to predict measurements. We're trying to, model the average of the lot. Now, your point in adding additional lots or having one lot is important as well, because again, this, but I'll stop there. You were trying to answer my second question.
Yeah. Well, Tim, actually, let's just stay in the first question, because you were saying now that we want a confidence interval. I do understand that we're not making predictions of specific measurements, let's say, but what about so going back to the model that was presented by Tobias, right, where we have these random intercept, random slopes. What about tolerance intervals on the tolerance intervals on the random slopes piece? That is not saying anything about specific measurements, but that is not a confidence interval either. So we're trying to say something about where a specific percentage of batches is going to lie, rather than what is a specific prediction. So now we're talking not about confidence intervals, we're talking about tolerance intervals. And prediction interval is basically just like a specific case of that tolerance interval. If you play with your parameters in your tolerance interval, you get a prediction interval. So I guess it is not clear to me that we need or we want a confidence interval all the time. We might need tolerance intervals, and not because we are focusing on specific measurements, but because we want to say something about a specific population of the batches that are coming from a consistent manufacturing process.
Absolutely right. It's what is it? A confidence interval on what versus a prediction interval on what? And the what is, to your point, it's the batch. So yes, I accept that.
Yep. And Tim, can you please remember me your second question? I am just.
Well, it had to do with it. In the traditional shelf life determination, we also account for, when it's appropriate, the intercepts, as it were, the different intercepts of the lots that are put on stability to forecast shelf life for a population of lots. How do you, with the Arrhenius method, incorporate the potential distribution or differences in intercepts?
Yeah, that's the other good question, Tim. I think so. I can probably share that I have seen I have seen the intercept being worked in different ways, and I think it also depends on how much risk you want to take or not. So what I have seen in some cases is that the intercept that is used to get the shelf life is not even informed by the actual data that you're collecting, but is more informed by the specification limits, in this case, the release limits. So you basically start your, let's say, your degradation at your release, which is, you can see that as a very conservative scenario, because you're thinking that, or you want to make sure that even if you release at the edge of the release limit, you are going to be okay in your shelf life. I'm not saying that's the best approach. I'm just saying that I have seen that type of approaches where you inform that based on the specification limit. Sometimes you inform that based on the tolerance interval that you build on your release data. So you have, I don't know, like maybe 10 batches of information, and you build some kind of like bound using that, and then you use that as your intercept. So I think those are just like two potential ways of using these, but like you said, it's not like we are going to be informing the intercept. On the other hand, Tim, there is actually one important point that comes with your question. And when I was presenting the slide with the differential equation and the two reaction rates, to solve that, I mean, at least when you look at this differential equation, to solve that, you need to have your initial conditions, right? Which in this case are the intercepts. You need to have information on the intercepts. That's going to be your initial condition. And I didn't even mention that in the slide, because to be honest, at least based on that one paper that I cite in there, it is not clear for me how they are informing the model in terms of the initial conditions. It seems to me that they are just maybe looking at the release data, and based on that, they are making an initial starting point. But it is not very clear for me how that specific paper is working with the initial conditions for learning the parameters of the model.
Thank you.
Tobias, I see a raised hand by you.
Yes, my hand jumped up in the first discussion point you had. I have so much to say to that. And I find it a really heated discussion. Why? For one example, should we include the measurement error in our shelf life prediction? In an ideal world, where the batches really follow our model, no. In a non-ideal world, which we are in, I would say yes. Like, I didn't do it in my presentation. We used the batch distribution. We don't include the measurement error. So this is kind of, I did this because this goes in hand with the ICH guideline. However, there is a paper from Kinlan, and he raises a really good point. In a non-ideal world, the measurement error, which we kind of estimate with our sigma for the measurement error, is not this variability, not only catches the measurement error. There are also other things caught where the model does not fit the reality. And in that point, it would be kind of more true if we would include that. This is kind of from the technical point of view. From the other point of view, from the patient point of view, I just think if I get a shot and the shot is of whatever, vaccine or whatever, or medicine, if that vaccine is good until 36 months, or the company claims it's good until 36 months, then I want to take it out of the fridge after 36 months, and I want to measure it, and I would like to be this one in spec. I wouldn't feel good if I know, if God knows that the truth is still in spec, but the measurement of this vial, which I take out and get into my arm, is out of spec. So from this point of perspective, I don't have a really good opinion on that, but there are also many arguments against not including it. As you can see, it was had to get out.
Well, in the sense of what we're trying to do, which is to describe things that have a known risk, a couple of things you mentioned, the risk can't be quantified. You can't quantify, well, what proportion is modeling error versus, unit to unit or measurement error. And in the case of testing at the end of shelf life, that is reducing the whole exercise to a single test value. So the risk profiles of all those things for making decisions about whether the lot is good or not are totally different. So they address different questions. I can't deny that a patient might want to know that that lot is good at the point that they receive their injection, but I would insist that the patient take all the data that was collected on that lot and do it, not just look at that last measurement, because it may fool them and they may not get their injection as a result. I mean, there's a risk that they may reject the treatment as a result of perhaps not looking at things more carefully. So we are in the business of helping them think about how to look at things more carefully, to understand the risk and to address the right questions. So, and you're right, it's a controversial discussion. Stan Alton and I have been involved in trying to debate the means versus individual values question for quite some time now, and it's a frustrating discussion.
I wanted to add something to this. Like, there's also another view on shelf life. You cannot only approach it via the batch distribution, where it cuts the acceptance criteria, but you can also derive from the samples, from the distribution, a shelf life distribution, and can take then from this the 5% quantile or something. Like, this is also a nice approach. We also looked into that. However, there are some inconsistencies, especially for the RIAS model, for the first random intercept, random slope model, where it makes it not equivalent to the approach with the intersection, which is a bit sad, because I would think having a distribution directly of shelf life, that would be the thing. That would be cool. That we do not need to talk about confidence intervals or intersections. Then you would directly have just a quantile of one distribution.
Right. Tobias, if I can comment on that, or actually question about that. I think at some point we were also looking at that second strategy for shelf life that you mentioned. And if I happen to remember, is when you have your rate to be like very close to zero, I guess that's when you have issues, right? Because I think you are blowing up the denominator. I mean, actually the number, because the denominator is going to zero. And but I'm thinking maybe on the other hand, because the values are closer to zero, maybe in some cases you can actually make or not you, but the expert, the subject matter expert, maybe they can make a reasonable assumption in there that that change is in fact very small, so it's not really affecting the shelf life you're thinking about.
Yeah, exactly.
Theo, I see your hand.
Yes, one comment and then a general question. So I liked your comment, Tobias, about that sigma, that error term. Sometimes we just say, oh yeah, it's measurement error, but I'm not quite convinced myself that it's just measurement error. In fact, if you think about how some of these things are collected, you're like, oh, actually there's some nesting going on or something like this. It's not capturing everything. So it's not just measurement, there's other stuff that's going in there too. So I appreciate that comment. The other thing is, I wanted to bring up, we're talking about novel techniques, novel methods. When I did my postdoc like almost 15 years ago, and I did it in functional data analysis, actually. And when I see data collected over time, and you have curves and you have trajectories, differential equations, like we had that presentation, the first presentation by Gang. I often go to like, oh, this is like functional data analysis, but but not, not quite. So my question is, have you ever heard of this, or and if you have you ever considered using it as a method?
I've never heard. Sorry.
Yeah, you've never heard of it.
No, but I could look for that. I'm
Yeah,
not sure what is functional data analysis. That's something is.
Yeah, and actually there's some ideas in functional data analysis where you could look at your trajectories and estimate parameters of your differential equations using functional data analysis techniques. So that's why I'm bringing this up. But you could do more than just that. But functional data analysis basically says the curve is the unit of observation and not the individual time points. You're looking at, the curve as the unit of observation.
Okay, that is super interesting. So basically you basically you're trying to fit a nonlinear or like time series of arbitrary trend that is not necessarily described by a differential equation, but might be just arbitrary trend. Is that how?
It could be
arbitrary.
Yeah, you basically use splines to do your fitting of the curves, and it could be general. There might not be a differential equation in the back there, but if there is a differential equation, you could use that as almost like a regularization to force your parameters to fit that differential equation. So there's a way of doing that. The main work was done by Jim Ramsey. He has a book on that, but then all his students also extended his work as well, and also in this differential equations domain.
That reminds me of a kind of popular technique called Gaussian process recently. I'm not sure if that is related or it's like different concept, but yeah.
I think it might be related. Yeah.
That technique is super popular in machine learning society right now. It's also kind of arbitrary a set of functional forms, and then you based on your existing data and try to fit that model. Which sounds like a very similar idea, but yeah, maybe it's different. I would definitely look into that.
Thank you.
Thank you, Theo. I see 2 more raised hands, and I think then we're already closing in on the end of the panel, but Jose, you would be next.
Yeah, no, Theo, that's a good point. I have used those techniques not in biotech, and I'm glad you brought them up. And as you were discussing, a lot of times you use some type of splines to fit those curves. But the whole point here, as Theo mentioned, is we're looking at the curve as the element, not the point. We're with this mindset where everything is a data point. Now you're dealing with the curves. But that also allows you to do very interesting things, because you can look at the rate of change, and you can look at the velocity and acceleration and do a phase plot where you plot velocity versus acceleration, and it will tell you stuff about how things are changing and how fast. So that's fascinating to explore. I had to step away and came back late, but I heard the conversations about some of the models. One of the things I've done in the past, not in this industry, is to feed a nonlinear mixed model with the Arrhenius. Okay, so some of the coefficients of the Arrhenius equation, you put some random effects, to do that. So that may be related to some of the questions. And in terms of the time zero and intercepts, when I've done these, one thing we did is to eliminate that, is to standardize by the value at time zero for each lot, for each individual. So all the curves essentially start at the same point, but they still have the same nonlinear behavior. So that's one way of sort of dealing with the different interceptions, standardizing by the time zero value, the whole lot, for example.
Thank you. Then Ben.
Yeah, we've talked a lot about the typical approach, fitting a different intercept, different slope model, and then testing the terms or whatnot, maybe reducing that down. But just throwing this out there for anybody, there's some that have suggested just from the outset, assume a common slope for the process, but fit the different intercepts, either random or fixed. And so I'm just curious if there's any thoughts about going that route, just kind of making some simplifying assumptions about the process. Doing so could, just simplifies the steps, the calculations, more simple. Would it be accepted? I don't know.
I'm surprised Stan's not speaking up because that's one of his positions. But speaking for him, and that I don't even pretend to do that, I'm not sure that that's scientifically logical for biologics which have complex degradation pathways. I think point that Stan makes is a chemical degrades in a certain way. And a small molecule degradation of that chemical is likely to be more predictable and or consistent than a large molecule, which is a heterogeneous population of polymorphisms that have different, each polymorphism, different characteristics that contribute to its degradation. I think that would be, assuming that every lot would be the same rate with the complexity of the degradation pathways of complex mixtures of molecules is a stretch.
Yes, Tim, let me just follow up on that. For small molecules, then, it's much easier to make that justification because the engineering and process controls for small molecules are extremely tight. And so we can assume that the kinetics is quite consistent from batch to batch, especially because we're controlling excipients, we're controlling the weighing operations, and so on. So the degradation in a small molecule can be considered to be like a property of that product, very much like it is for any molecule.
Okay. Well, thanks for the thoughts. Just had to throw it out there.
Thank you very much, Ben. So looking at the time, we actually reached the end of our panel discussion. Maybe when I just look back, I think we can say that if you put two CMC statisticians in the room and say stability, they will have a lot to talk about it. That's something that I always encounter when the topic goes to stability. It's a never-ending story, but always a funny, lively, vivid discussion. With that in mind, I want to thank everybody on the panel for great presentation, for great discussion, everybody who contributed to this panel discussion. But of course, I want to thank also everybody in the workshop again. But to do this properly, I would hand back to Julia for a bit of a workshop summary.
Okay, thank you. And yes, thanks to everyone. Really 4 days of excellent presentations and really interesting and lively discussion. I know I took away a lot from this meeting. And I do want to thank in particular our IABS colleagues who did so much work behind the scenes to make this such a smooth running virtual meeting. Abby and Maddie have been with us all year working to set this up. I think the breakout rooms are just amazing, and there's never a hiccup. It goes so smoothly, helping everyone with their preparations, making sure that the slides went well and the audio visual worked. And the thing I sometimes forget is they're doing all of this from European time zones, which I know many of our presenters and participants are also in Europe. And you're staying up very late to be part of this conference. Well, maybe for you all, maybe you're night owls and it's not late. For me, that would be like already past my bedtime. So I just want to say a huge thank you to IABS for the support, and also to our sponsors. I believe it was Merck, Genentech, and Pfizer this year, and we really appreciate that support, as well as Maddie and Abby. Thanks again for your support throughout the year and across all these time zones. Let me see if I can share my screen. I'm going to try. Can everybody see this a slide now?
Yes.
Great. I'm going to try. Okay. So I did my best to pull together some highlights from the workshop, and I want to give a big thanks to John and Roja and Stan for taking very good notes. I tried to condense these into 1 page per session, which you can imagine is very difficult to do. I can't seem to advance the slide, so I'm just going to do it the awkward way. All right, here we go. Yeah, so this is boiling down some of the things that I'm going away with from this meeting. Our keynote, Valentina from CEPI, it gave us a very interesting perspective on the context of our work. So CEPI looking to accelerate the development of vaccines and ensure better access by global populations. It was very interesting. And then when she got into the technical content of her talk, it really made me think back to the chemical industry, where we did a lot of round robins, and very encouraging to see that in spite of the challenges with COVID and its ability to change, that there is progress being made towards establishing correlates of protection. Then we moved into the main session for the day. Applications of statistics, CMC statistics to new modalities. Is a great talk by Rick, as always, on statistical approaches for CMC comparability, especially as applied to gene and cell therapy. Nice example, and it was a real takeaway for me, the importance of intra-class or donor variability in designing those tests and studies for comparability. So I think all 3 of the talks in this session, I'm hoping to get my hands on them and use them as tutorials almost. Oh, and I forgot to mention that all the talks from this conference, which were cleared for sharing by the companies involved, those will be available on the IABS conference website beginning next week. So of course, you all know not every company will allow slides to be shared, but those who do, the slides will be available. And I'm certainly going to look for these. The next talk was John and Catherine on Bayesian modeling for comparability in a novel for tissue engineered human acellular blood vessels, really cool application. And an example of clinically relevant attributes too, and using Bayesian methods for those. And then Jose, now with Kite, and another great talk on monitoring cell viability and bringing in techniques that were established for reliability, and also Bayesian modeling to calculate tolerance bounds. So all 3 of them quite practical and innovative at the same time. Really great. Okay, going back to my clunky way of doing this. And I actually, the scribes sent along really great summaries of the discussions too, but I think in the interest of time, I'm going to focus on the talks in these closing summaries. So from day 2, that was an experiment for us, seeing how it would go when we had a day that was really devoted to a couple of things. The main focus of it being to expand the reach of all of us, especially through this conference, to connect more strongly with the data scientists we may already be working with, and to bring along a new generation of CMC statisticians, because I'm sure all are familiar with how difficult it is to find people working in this area, as we're such a high demand, just not enough supply. So building these bridges with data scientists is one way we can work on that. Great kickoff by Sean from Biogen. The only point I would take issue with is that Sean mentioned that CMC could borrow approaches from preclinical, biomarker, clinical medicine. And I actually think this is a two-way exchange, where I have seen that some of the things we are quite adept at in CMC are not so familiar in sometimes preclinical, biomarker, and clinical. And so I think this is a two-way street. Then a whole series of really informative talks about people's pathways through to CMC statistics or adjacent fields. So Brandy from Merck talked about the need for us to show value. And I think this applies not only for data scientists or statisticians, I mean, it really applies equally. And then Tara from Synalostats reminded us all that we should be focusing on the risks and decisions that we need to make using statistical methods, and less emphasis on the jargon and Greek letters. I think it lines up nicely with the previous Brandy's comment about showing our value. And Jonathan from Resolute brought up the impact of his motivation for a career in our area because of family experiences with cancer. It's certainly a big motivator for me as well, and in working in accelerated fields. But then he was fortunate to find a path in CMC statistics, which not only allowed him to work on important health issues like cancer, but aligned with his personal professional interests. And then Jeff from Data Farm, his consulting company, really interesting perspective on how he came to statistics as a tactile practitioner, likes to get his hands dirty, and is looking for methods that are effective. Whether or not they're perfect is secondary. And then Sarah from Bristol Myers Squibb, with an early career story of how she, as she described it, stumbled into CMC statistics, but she's been noticing some of her data science colleagues seem to be better at storytelling, and something for us to learn there. And I would second that as well. And last but not least, Sang-Wook from BMS also said how collaboration is key to success within quantitative and cross-functional teams. Absolutely. I think that part of the whole focus of this conference is on the context in which we apply statistics. And so collaboration and communication are essential to be successful there. Then I will go to here we are. Yeah, sorry for the clunkiness. Yeah, so then day 3, just yesterday, another great session on regulatory opportunities and advances in CMC statistics. We started off with Mats from the Swedish MPA, and he gave a really great overview of progress that's being worked on for stability guidelines as well as specification setting, and highlighted the EMA Prime Toolbox, which is a great set of, it's really a great summary of many of the tools that many of us have been advocating for some time. And, you know, just another great update from Mats. And there was, I will mention some of the discussion that followed was, boy, this is a great approach, the EMA Prime Toolbox. Can we use it for products other than Prime? And he encouraged those of us working for sponsors to approach agencies as early as possible with that idea if we want to do that. And then Anne from Quantix with another excellent talk on total error in bioassay. Really, really interesting. Oh, and one thing which I found especially intriguing is the connection to the CPM process capability metric. I think I've got some reading up to do. That's something I used to be familiar with quite some time ago, the Taguchi CP index, but time to look at that again. Then we went to continuous processing for vaccines with Laurent from GSK, with a great example from adjuvant manufacturing, and highlighting some of the challenges for continuous processing. So batch definition is certainly one that needs to be overcome to implement in the regulated environment. But then some useful things emerging, ICHQ 13 providing guidance, and CQA real-time predictions available from digital twins and hybrid models. Then the last talk yesterday was on progress and opportunities from the CMC Bayesian Scientific Working Group by Paul. And Paul's very brave, I think. He's from Lilly, and there's always a lively discussion when we get into Bayesian applications, but he gave an excellent summary. There was a lot of useful references in his talk, which I hope he'll be able to share. It was just too many to include in this brief summary, but certainly you may make a Bayesian anatomy yet, because the application to stability is something I'm finding very intriguing. So lots of good references there from Paul's work. And then finally, did I lose it? Oh, shoot. Oh, here we go. Yeah, so I didn't have time to get notes together for today, but hopefully this is also fresh in our minds that I won't need to do much reminding. Another great session with applications of new CMC statistics methodologies. And starting off with Mark and Gong from Moderna, giving a case study on continuous manufacturing, applying QBD, PAT, and digital twins. And then a couple of talks on stability using Bayesian and other Arrhenius approaches. The first one by Tobias from BI, and then Christian from Genentech. Really, I mean, we had to kind of shut down the discussion. It was so interesting, I think, for everyone still on the meeting, and really looking forward to maybe interacting with you in the future at future conferences in these areas. So that is a very brief summary of all the topics we covered in the last 4 days. I want to encourage you all for a couple of things. First, I hope that you will consider joining us again next year for the 2023 workshop. That will be co-chaired by Laura Pack and Timo Baylor for next year. And the dates are still to be determined, but it will be sometime in the fall, most likely. But they are going to work through a choice of dates as well as venue. We all, I think, hope that maybe we could meet in person again in the future. Although in our conference committee, we've often remarked on how well this particular conference works virtually, because it makes it quite possible for us to join together without, days of travel for some members of our group. So please stay tuned for more information on next year's conference. And if any of you are interested in becoming involved with the Scientific Organizing Committee, I'm sure that Timo and Laura would be happy to connect with you and explore that possibility. let's see. Okay, let's see. I thanked Abby, Maddie from IABS, and I certainly want to thank the Scientific Organizing Committee, in particular my chair, Christy Griffiths, and it's really been great working with all of you. I thank you for your many great ideas and your creativity in experimenting with new formats for this year's workshop. And I hope that many of you will stay engaged through to next year. I'll just open it up for any last comments or questions before we close. Oh, I did forget one thing. Please fill out the survey that Marlene is sending around. And thank you to Marlene also, who has supported this conference. Yes, please fill out the survey. We are always trying to improve from one year to the next. And we will hope to see you all again next year. And that concludes the 2022 IABS Statistics Workshop.
Thank you. Thank you.
Thanks.
Thank you so
Thanks,
much.
everyone. Thank you so much.
Thank you.
Thank you.
Thanks very much. It was great.
Oh, thank you, Tara.